Total RBC count. The counting of blood cells manually with help of microscope is not possible. Therefore, to count the cells, blood is diluted and placed in a special type of counting chamber. This technique is called as hemocytometric counting. The cells often counted by this method are red cells, white cells, platelet, and eosinophil. Red cell count. For total red cell count, following equipments are required: RBC diluting fluid, RBC pipette, hemocytometer, microscope, and a blood sample. RBC diluted fluid. The RBCs are about 5 million cubic millimeter of blood. The counting this much number is high impossible. Therefore, the blood sample is diluted with the help of RBC diluting fluid. It fixes the preserves RBC. It fixes and preserves RBC. It is isotonic to RBC. The following two types of RBC diluting fluid are commonly used: formalin citrate diluting fluid. Composition: trisodium citrate 3 grams, formalin 1 gram for 1 ml, distilled water 99 ml. This diluting fluid is cheap and commonly used. Hames diluting fluid. Composition: sodium chloride 0.5 grams, sodium sulfate 2.5 grams, mercury chloride 0.25 grams, distilled water 100 ml. The RBC dilutive fluid preserves hemolysis, prevents hemolysis, and removes unwanted blood cells. RBC pipette. The RBC pipette is graduated to give the dilution of 1:100 or 1:200. It has a bottom marked with 0.5 and 1, and the top is marked with 101. It has a round bulb containing a red bead. The rubber tube is attached to the top for sucking. The blood sucked up to 0.5 mark gives us a dilution 1:200. In case of anemic patient, it is sucked up to 1 mark to give dilution 1:100. After blood, the RBC diluting fluid is sucked up to 101 mark and mixed gently. Hemocytometer. This is a chamber used for cell counting. The generally improved Neubauer chamber is used. Other chambers like Burker and Fauch's Rosenthal may also be used. The Neubauer chamber has ruled area of total 9 square millimeter and the depth is 0.1 millimeter. The central one square is highly ruled. The central square is divided into 25 squares. Each square is further subdivided into 16 small squares. For RBC count the cells in each corner and one central square, that is 5 square. As each square is further divided into 16 small square, the area is to be counted is 16 into 5 is equal to 80 small squares. Blood samples. The capillary blood or EDTA anticoagulant blood may be used. Principle: the blood sample is diluted usually 200 times with the red cells diluting fluid. The diluted blood is placed in hemocytometer. The cells are counted under proper magnification over the specified area of hemocytometer. Known factor or number of cells counted, volume of the fluid inside the counting chamber, dilution of the blood. With help of these known factors, the number of RBC cubic millimeter of undiluted blood can be calculated. Procedure: assemble all equipment. Draw the blood directly from the finger or collect sample into the RBC pipette up to 0.5 mark. Wipe off the tip of the pipette to remove extra blood if present. Then immediately draw up the diluting fluid up to 101 mark. Now rotate the pipette gently so that the diluting fluid get mixed properly. This will give the dilution 1:200. Place the cover slip in position over the rolled area of the chamber. Once again, mix the solution thoroughly by rotating the pipette. Now apply the slight pressure on the rubber tube of the pipette so that the second drop of the fluid is in hanging position. Touch the tip of the pipette hanging drop against the edge of the cover slip. The angle between the pipette and the cover slip is 45 degrees. With this process, a chamber gets filled with the fluid. This is known as a charging of chamber. The care should be taken that no air bubble is present inside the chamber and there is no overfilling beyond the ruled area. Leave the counting chamber as it without disturbing for about 3 minutes. This will allow settling down the RBC. Place the chamber on the stage of the microscope. Adjust the light and ruled area. Make sure that the distribution of RBC over the chamber is uniform. Now count the RBC in central square. The central square is subdivided into 25 square. Out of 25 square, count 4 at each corner and 1 at center. Each of these 5 square is subdivided into 16 squares. Thus, RBC are counted in 16 into 5 is equal to 80 small square. In case of marginal cells, count the cells on L line, either right and left or left and upper margin. Make this total of the cell counted in 5 square and repeat the procedure to another side of the chamber.